Well, if you remember, within the previous videos we talked about the fluorescence microscopy and the use of fluorophores for these imaging techniques. We mentioned that different fluorophores with different colors can be functionalized with the groups which have certain specific affinity. Affinity means the attractions towards different parts of the cells. For example, we can get the red fluorophores and equip them with the functional groups which have the affinity towards the cell membrane. So as you see here, when they are introduced, they will stick all around the cell membrane. And on the other hand, we can have our foreign objects, the nanomaterials or anything, the proteins, and we can equip them with the green fluorophores. So, by getting these pictures, we can see whether the nanoparticles, the proteins, have been internalized or not. But we mentioned here in this picture that we have different endocytosis mechanisms, and each of these have their own different cascade of chemical reactions. We also talked about the use of dynamine, which is used for the closure of this vesicle. So, the final step of the internalization process. In order to understand which of these mechanisms is preferentially used by the cells, we can use some materials such as dinosaurs or these materials to see which of these mechanisms is used more by the cell. Okay, dinosaur is a sort of inhibitor which inhibits the dynamines to close that vesicle. So by using different inhibitors to inhibit the, for example, caveolin mediated endocytosis, the clathrin mediated endocytosis, we can see either by inhibiting one mechanism, the other mechanism is still responsible for the uptake of nanoparticles or not. Okay, for example, if we use the dinosaurs to inhibit the clathrin mediated endocytosis, and again the population of the nanoparticles which are internalized within the cell does not decrease that much, we realize that this is not the clathrin mediated endocytosis responsible for the internalization of the particles inside, but this is the caveolin mediated endocytosis. But always remember that we cannot 100% inhibit the internalization of the particles or objects within the cell. If we inhibit one mechanism, the cell will definitely come up with another internalization mechanism to do what it wants to do. Okay, let's have a look at this picture. If you remember before, we told you that within different steps of the internalization, we will after that go into the early endosomes and the late endosomes, and there are some proteins which will be recycled and some proteins and enzymes coming here within the late endosome to degrade the materials. Okay, if we use the fluorochromes which are having specific affinity towards the different proteins in the late endosomes, in the early endosomes and lysosome different steps, we can see when and where the populations of these specific proteins is going to be increased, and we can see the kinetics and also the dynamics of the degradation process within the cell. Okay, for example, these are different proteins, the RAP7, RAP5, 9, 11, which are present in different steps of the degradation process, And as you see here, their population, their concentration is changing with the matter of time, and by studying these diagrams, the kinetics and dynamics of the degradation process will be decoded. This study shows the dependency of the internalization by the cells on the concentration of the particles which are around. Okay, by increasing the concentration, of course, the internalization will increase. As you see here, we have the 10 nanomolar, 3 nanomolar, and 1 nanomolar. And as the concentration is increasing, the internalization is also increasing. But it will happen until a certain concentration. And after that, we will reach into a sort of a plateau, and we will reach into saturation, at which any further increase in the concentration is not going to be useful to increase the population of the internalized nanoparticles within the cell.